So, a little about today's presentation. We're going to go over some experimental best practices for single cell for those that might be new to the workflow. And then there's also the workflow overview we're going to touch on, along with some sample prep best practices for Illumina single cell. We also have resources for those of you that want to look at all of the different documentation that we use to put together today's presentation for those of you that download the slide deck later. So I guess the best place to start is, what is single cell sequencing? Well, it's one of the fastest growing next-gen sequencing methods, and at least over half of next-gen sequencing researchers are using single cell methods at this time of some variety. It's essentially, at its core, a technology that is used to understand individual cell populations in complex tissues or organisms or in diseases at a DNA and RNA and epigenomics or a protein level. And so there's a wide variety of applications of biological information that we can get at a single cell level. Now, Illumina single cell uses PIPseq technology, which a PIP stands for a pre-templated instant partition, which is comprised of these hydrogel bead cores with a partition that is formed around it. And the hydrogel bead has a PCR handle with a cell barcode and a binning index here in place of what we would typically see a UMI, or a unique molecular index, in many single cell technologies. We have a binning index instead, and we will go into a bit more about that in the workflow overview. There's then a poly-T capture moiety for capturing those polyadenylated mRNA molecules. And so we can see that what we have here with the general overview of this type of workflow is to capture polyadenylated molecules and reverse transcribe it into cDNA. So Illumina single cell is using or creating monodispersed emulsions by vortexing. So we have here in the upper left, we have a mixture of pips and cells, or our sample, in the bottom in an aqueous phase. And in the top, we have our oil, or our partitioning reagent. We vortex, and then after brief vortexing, we should end up with single cell droplets with our sample. Now, this workflow is done with minimum amount of instrumentation. The starter kit is relatively inexpensive. It doesn't take up much of your bench space, but it is engineered to cause or generate a uniform droplet generation by vortexing process. And so this proof of concept was demonstrated by the Fluent Biosciences group in this 2023 paper that's cited at the bottom of the slide, for anywhere down to 200 cells in a PCR tube up to 1 million cells in a 50 ml conical. Now, the existing products that Illumina single cell enables right now are at the 2,000, 10,000, 20,000, and 100,000 cell kit sizes that you can see here. And so the scalability of this technology of droplet generation by vortexing is incredibly powerful. So we are going to go into the workflow in greater depth further on, but Illumina single cell, when we're talking about the workflow, we're talking not just about that droplet generation by vortexing to capture our single cells. We're also going to lyse those cells and capture the mRNA on our poly-T capture moiety. We're going to reverse transcribe that material into cDNA and amplify that cDNA. We're then going to do a library preparation in order to add our Illumina-compatible sequencing indexes to run for sequencing, and then do analysis in Illumina-connected software. So the first two steps of this workflow, capture in PIPs and lysis and mRNA capture, is about 10 minutes of hands-on time, a little less for experienced users, to get to the first stopping step of the workflow. And key steps in the Illumina single cell workflow that we're going to touch on today are going to involve high quality sample preparation, such as high viability cell suspensions or nuclei with a high RNA integrity number, or RIN value. We're also going to touch on the sample wash step and its importance and some of the best practices for cDNA and library QC and some typical expectations of some example traces. So let's get into single cell experiment best practices. So let's say you're new to single cell. You've never done this technology before with us or with any of our competitors. Well, where do I begin? I begin by going to the literature for different cell or tissue preparation methods for my sample type, and to see if anyone else has done this with this tissue for single cell sequencing. So first I go to the literature and see what other people might have done. And then I'm going to learn what I can from those protocols, incorporate all of the PIPseq specific sample prep recommendations from Illumina's customer training packet into the chosen protocol. And we're going to cover what some of those recommendations are here today in today's conversation. There's also some short video tutorials linked in the customer training packet, but they're also on Illumina's website for the Illumina single cell product as well that show proper key PIPs handling technique for all of the bead handling portions of the PIPseq workflow. And then finally, plan a pilot study. I'm going to sound like a broken record today with suggesting these pilot studies, but a pilot study is so important and key to an informed experimental design to ensure that when you scale up your sample number or cell number to a bigger kit size, that you're confident that you'll be able to meet your experimental objectives. So let's talk about adequate cell numbers and which kit size do I choose for my application. Well, it has something to do with sample heterogeneity and the population frequency of your cell type that you're interested in. For most people working at the T2 kit scale that looks at about 2,000 cells, maybe a little bit more if you're lucky, or you're experienced, you got homogeneous or enriched sample types in most of those situations. Most customers are working with some sort of cultured cell type or enriched cell type by flow sorting or some other sort of enrichment process, because cell type heterogeneity here is going to be difficult to cluster with populations that are less abundant than 20% of your total cells. If you're looking at more heterogeneous tissues and you want to get an idea of cell subtype heterogeneity, T10 begins to be more appropriate, where you're looking at 10,000 cells. Cell subpopulations at 10% or greater can be resolved reliably. But if you're looking for those rarer cell types among their native tissue environment and you want to see everything in the whole tissue without any sort of enrichment, well, the T20 or the T100 might be much more your speed or your size for your experimental purposes, where you can see those rarer cell subpopulations in the total tissue. With these increased numbers of cells in a single reaction in a single experiment. So many customers at the T100 scale, if they're looking at that 1% cell subpopulation, but they don't want to spend all that extra money at sequencing to look at the full tissue, they might do a flow sorting or an enrichment workflow to enrich those cells and put them into a T2 instead to save some money in their experiment design at the sequencing level. So when it comes to experiment design, we're going to, again, emphasize the importance of pilot studies. You should do maybe 1 or 2 samples to get used to the workflow prior to doing an actual experiment that you're hoping to generate data from. This is incredibly important when you're working with a new sample type that you have not prepped for Illumina single cell before, and you need optimization of the upstream sample prep before you're going into Illumina single cell. That old saying, garbage in, garbage out, right? So we want to make sure that a pilot study is ensuring that our sample quality is good quality to go into the workflow. Because if we put a low quality sample in with struggling viability and like the 60 or the 70% viability, we should expect a linear decrease in the single cell metrics with a linear decrease in cell viability. So that's another reason why we want to hit that 90% or higher viability. So a T2 can be a great economical way to do an inexpensive and small pilot study just to see if your sample prep practices are appropriate and are working for Illumina single cell. Whereas a T20, if you're looking at larger cell populations and and different cell subpopulations at a larger scale, a T20 might be the appropriate pilot study for you before scaling up to a T100 if most of your experiments are going to be done at the T100 scale. So we do want to remind our customers about best practices with working with RNA. This workflow is an open system. We're not in an enclosed microfluidic device. So that means your samples, while processing them, are more exposed to exogenous RNases from the environment and people. So that means clean your workspace in your pipettes, maybe a PCR hood if you have access to that, like a dedicated workspace for your molecular biology. And of course, just don't touch any of your single cell reagents or equipment with your bare hands. Most exogenous RNases come from skin. So we just don't touch any of your reagents, any of your pipettes. Please don't touch things with your bare hands. Use your PPE in the lab. And of course, we have a reminder here for some standard cleanliness practices for keeping your workspace clean. So it's also good to separate these types of RNA workflows in your pre-PCR and your post-PCR workspaces. So everything done with RNA and setting up your reactions to generate your cDNA or to generate your library should be done in the pre-PCR space, whereas like your magnetic bead cleanups and your QC should be done in your post-PCR workspace. And this is a good reminder so that best practices, we prevent cross-contamination of past experiments and amplicons from other samples with experiments from ongoing or current samples. So now that we've covered best practices, let's get into the workflow overview. So we showed this slide already and gave a bit of an overview of what we're doing at a high level at each of these steps along the road, and that we only have about 10 minutes of hands-on time to reach that first stopping step. Well, let's get into this workflow in the top part that we see at a high level in the top part of this slide and cover what's happening with our samples. Well, for cell capture and lysis, we have our mixture of pips and cells or nuclei, whatever our sample is, with our partitioning reagent in the top phase. We vortex and we generate our single cell droplets, and then we're going to have some excess partitioning reagent that needs to be removed before we add our lysis reagents. After we use these lysis reagents, they're going to go into the PIPseq or Illumina single cell dry bath to be incubated for about an hour, 90 minutes. And this, once the samples are put in this dry bath, is the first stopping step of the workflow. So it's just a couple of pipette additions, a removal of some excess reagent, and a vortexing step before the user reaches the first stopping step of the workflow. Not too bad. And it's especially powerful for those that have lengthy sample prep workflows from like long tissue dissociations or enrichment processes. Now, after we have put the cells in the dry bath, they are lysed inside our droplets. So we should have our mRNA hybridized to our poly-T capture moieties inside the droplets. We're then going to break these emulsions open and wash the pips. So we're getting all of that aqueous phase prepared for reverse transcription, where we do our first strand of cDNA synthesis that covalently extends the first molecule of cDNA off of the PIPseq bead itself. So we then do another set of washes to get all the primers and enzymes out to reset the environment for a new PCR reaction for whole transcriptome amplification, which is going to be for 5 PCR cycles only, and it must be 5 cycles, no more and no less. And this is to create 15 soluble copies per captured transcript with that one bead bound that was formed during reverse transcription. Now, we need to keep that whole transcriptome amplification cycle number the same for our transcript counting method using the binning index, and we will get more into that in some upcoming slides. So this cDNA amplification is necessary for this 15 soluble copy per captured transcript assumption for the analysis process. So that 15 soluble copies of each transcript, however, is not a high enough yield for QC. So we need to set up a secondary QC cDNA amplification to over-amplify the cDNA so that there's enough of it so that we can see it on a tape station or a fragment analyzer or a BioA. We want to see that the average size is above 500 base pairs. And if your average size is below 500 base pairs, you're going to have to ask yourself, how can I improve my sample prep to prevent mRNA degradation? Is there a more RNase inhibitor I can add? Is there a step that I can speed up or do more quickly? And we have a link here to some example traces on Illumina knowledge base. Now, during the library prep portion of the workflow, we're going to use that entire 5-cycle cDNA product and put that into the library prep. So we're taking everything from that 15 soluble cDNA copy cDNA product into the library prep. We do not want to use the QC cDNA. It will cause the analysis to fail. So we take the entire 5-cycle cDNA product and we put it into library prep, which begins with fragmentation. This fragmentation is essential for generation of our IMIs, or our intrinsic molecular identifiers. Now, there is a paper here for how these IMIs were developed scientifically by the Fluent group here. And then we also have the use of IMIs in place of unique molecular identifiers, or UMIs, that we see in many other single-cell workflows. So what happens here is we have a fragmentation occur where we cut the cDNA. And this unique cut site, when combined with the binning index, is how we're able to bioinformatically demultiplex the transcript number back to an original number of transcripts captured, or molecules captured. So fragmentation generates this cut site, which is essential for intrinsic molecular identifier generation that is used for Illumina single cells transcript counting analysis. After we do fragmentation, we do adapter ligation and sample indexing PCR. And after that sample indexing PCR, our library should look something like this on the bottom, where our cDNA insert is flanked by the Illumina single cell barcode structure and a P5 moiety, as well as the 3' end of the amplicons having our I7 structure as well. So when we do our library prep, we're going to go to make sure that we are pooling appropriately for how many samples we're going to sequence together. And so please refer to the adapters pooling guide here that we've linked in this slide. So we have 8 UDIs, or 8 unique dual indices, that are premixed that come with the kit. And if you need an increased number of plexity or need to sequence more samples in a single lane for some reason, we do have the Illumina single cell UDI 96 sample plate available for increased plexity. Now, best practices for library prep and QC would be, if our average size is greater than 370 base pairs, but looks like this nice normal distribution here, that's perfect. That's green. We're going to sequencing. If our average size is somewhere between 350 and 370, that is the yellow zone. That is proceed at your own risk. You may be able to get some useful data out of it, but it may not be the most efficient at being sequenced, or there may be some lossiness in the data itself. And then if your average library size is under 350 base pairs, do not sequence this. It's going to be a waste of your time and your money. This is going to be needed to be troubleshooted. So anytime you have an average library size where it's less than 350 base pairs, we need to examine if there was mRNA degradation that occurred and how we can improve our sample prep in the upstream to prevent that degradation so that we can get these library average sizes larger. If the average size is trailing off and looks way too large, then that could be indicative of a fragmentation issue. And in those instances, it may be possible to go back and regenerate libraries from the PIPs if possible. So when we are doing a pilot study or we are trying a new sample type or we're new to Illumina single cell, we would recommend doing deep sequencing. So what we have here on this slide is our new customer sequencing recommendations. And this is what we would do for not just new customers, but experienced customers pivoting to a new sample type or a new species. Start with this deep sequencing of 20,000 reads per input cell. This is to help determine the capture efficiency of your new sample type and the efficacy of it in our workflow. Once you have determined your capture rates and seen what the specs of your sample are like in this workflow and have done your pilot study, there is a less deep sequencing recommendation for known samples once you've done that pilot study for 20,000 reads per expected cell. So instead of doing 100 million reads for the T2, you're doing 40 million reads, for example. So this is another reason why that pilot study is so powerful, is once you get that information about your sample and how it behaves in the Illumina single cell workflow, you can start scaling back on the sequencing and don't have to sequence quite so deeply. So those pilot studies with small sample numbers are really nice for getting the specs on your samples. When we go to sequence, we have these final library loading recommendations for each of these Illumina sequencing instruments, with at least 1% Phi-X on the lower capacity instruments and 2% Phi-X on the NovaSeq X. The library structure is a read 1 of 45 cycles and a read 2 of 72 cycles. If you are doing a 100 cycle kit, you can squeeze 137 cycles out of that kit when you do the 10 cycles for the i7 and the i5 on top of that 45 and 72 cycles for the read 1 and read 2. But if you're doing a larger capacity kit, like a 150 or 200 cycle kit, we would recommend that you add those additional cycles to read 2 so that you can read deeper into your transcript. If you add those additional cycles to read 1, you're going to read into the poly-T, and that's going to reduce the efficacy of the sequencer with that reduced sequence diversity through that region at those cycles. So please add the additional cycles to read 2. So once we've generated our sequencing data, what do we do to analyze? What's our Illumina connected software? Well, our Illumina connected software for Illumina single cell analysis is Dragon, and it is a free analysis software with an on-premise Dragon server. It's also free to launch in the BaseSpace app as well. And it's free to run even in BaseSpace also. The difference in where the free and payment delineates is storage. So if you're working with a free trial or a basic account on BaseSpace, you have free storage of up to 1 terabyte of data. Once you go over a terabyte of data, you're subjected to that data deletion policy. If you are a subscription account with BaseSpace, then you have that free storage up to 1 terabyte, and anything over 1 terabyte you will be charged for. And we do have a link to the Dragon Illumina PIPseq single cell RNA guide here as well. So what does Dragon do for for its IMIs, or the intrinsic molecular identifiers that I talked about earlier? Well, it's using those first 12 bases of read 2 as the IMI, or that unique cut site. So it's combining those first 12 bases of read 2 with the binning index to do our molecular demultiplication so that we can do our demultiplexing of our molecule counts. And so Illumina collaborated with Harvard University's genomics core to compare GEM-X to Illumina single cell. We used the T20 kit and used the Illumina platform internally here at Illumina using Dragon v4.4. And the 10x samples were run at Harvard by their core lab, and they used Cell Ranger version 9. The goals of this project were to remove bias by performing the prep of the competing product outside of Illumina and to downsample as needed to normalize the number of cells sequenced to be equal across both technologies and the sequencing depth of the number of reads per cell to be matched or normalized between the two technologies as well. We also sequenced both samples to 150,000 reads per cell, which is way over sequencing. So we sequenced incredibly deeply on this comparison study to allow both technologies to perform as well as reasonably possible. So in that comparison, one of the things we wanted to highlight was Dragon's runtime is much faster than our competitor, with some of these runtimes being 3 hours, 2.5 hours, on analysis times that would otherwise be anywhere from 8 to 24 hours with some other pipelines, or the Cell Ranger pipeline in this instance. So Dragon is incredibly fast. And when doing cell type identification, you can see there's incredibly good concordance between the Illumina single cell UMAP here and the 10x Chromium GEM-X UMAP as well. When we look at the violin plot, the empty circle is the Illumina single cell data, with the filled in circle being the 10x GEM-X. And when we get to these conventional dendritic 1 cells, we see that this cell type is only visualized in the Illumina single cell platform along with HSPCs. So that's the workflow from beginning to end. But now let's talk about what you, the customer, bring to the table, your sample, and how can we best prepare that sample to put our best foot forward with Illumina single cell. So we've already talked at a high level about what the entire workflow is and where these stopping steps can play into the researcher's hands and flexibility. But what do you need to do to bring this workflow the most power to your experiment? And that is to bring a high quality cell or nuclei prep to the Illumina single cell workflow. So what are we doing to bring that high quality sample to the workflow? Well, first, we need to decide if we're working with cells or nuclei. These could be derived from any number of sources: cell culture, tissues, blood collection, you name it. When it comes to single cell sequencing, we need to consider that any number of these cells could be from any number of sources. But if they're larger than 80 microns in diameter, we should consider nuclei isolation. That's just one of the physical constraints of the droplet formation by vortexing, is cells that are larger than 80 microns in diameter just are less likely to be captured in the same droplet as a PIP hydrogel bead. So you should consider nuclei isolation if your cells are particularly big. And then single nuclei sequencing enables customers to do frozen tissue. So cells you can dissociate from fresh tissue, but if you put that tissue in the freezer, you've now started the first step of nuclei isolation from tissue as a freeze thaw. So if you've put that tissue in the freezer, you now have to go the path of nuclei. You cannot isolate cells. Those cells are not going to be very happy if you try to dissociate them from frozen tissue. It also enables large cells for cells that are larger than 80 microns, and it enables cells that may have cell walls, like plant cells. So there is the Illumina single cell nuclei isolation kit that Illumina does sell, but there's also a vast number of alternative workflows, and we are going to discuss today which should you choose to put your best foot forward for your application. But first, let's talk about cell suspensions. And if our cell suspension doesn't meet that 90% viability recommendation. Now, that 90% viability recommendation is consistent across like every single cell product. So it's not just us that recommends that. But there are a number of dead cell removal kits out there on the market, and we've heard from customers in the past that the Academium Life Sciences kit has worked for them to improve their viability prior to Illumina single cell, as well as the dead cell removal kit from Miltenyi Biotech. So the Academium one requires 500,000 cells to start, the Miltenyi one requires 1,000,000 cells to start, and they have two different mechanisms of removing your dead cells from the suspension. And there are other commercially available kits, but these are two examples of ones that customers have reported being happy with in the past. So but if you are working with an incredibly rare cell type that you can't get up to 500,000 cells to input for this viability cleanup, you might be looking at doing like a live dead cell sort. So let's think of nuclei now. So for cells, we discussed how we could clean up that viability with some example commercially available options. But with nuclei, we have a decision tree to consider. We have the Illumina Nuclei Isolation Kit that has been validated for frozen mammalian tissue. Now, that's been tested internally and done by our R&D scientists with mouse brain, heart, liver, and diaphragm, those four tissues. If you are working with a different species or a different tissue type, then this kit may require some optimization for an unvalidated tissue type or for a cell suspension, since this workflow is optimized for frozen tissue, not cell suspensions. So this is a kit that has a limited, narrow scope of focus for what it can do out of the box as is written. Otherwise, it may require some pilot studies to optimize lysis incubation time, number of homogenization strokes, and things of that nature for a new tissue type. Now, if you don't want to do this optimization uh, for nuclei prep with Illumina's kit for your tissue type, there may be an alternative nuclei isolation workflow that you're already familiar with, you have developed in your lab, or is a commercially available kit out there on the market. Now, it may be necessary or it may even be optimal for you to use this alternative nuclei isolation workflow over Illumina's kit, especially if it's for a species or tissue type that has not been demonstrated with the Illumina nuclei isolation kit. However, if you're using an alternative kit, we recommend that you incorporate all of Illumina's recommendations for alternative nuclei isolations. We have a lovely knowledge base article that reviews all of those recommendations up on Illumina Knowledge, and it's in the training packet as well. So please review those recommendations that should be incorporated to put your best foot forward with your own nuclei prep for Illumina single cell. And of course, pilot study. I said I was going to sound like a broken record. Here it is again, pilot study. Trial that nuclei optimization for that tissue type if it's a newer unvalidated tissue type, or optimize any additional nuclei handling steps such as staining, labeling, flow sorting. Sometimes the nuclei isolation isn't the last step for some of our customers before they are ready to put it into the single cell workflow. So many things that you can do to test your sample prep of your nuclei quality is to remember some of these key points. And that is to move from cells or frozen tissue to encapsulated nuclei in the Illumina single cell workflow in the PIP droplets as quickly as possible. If we wanted to test our pilot study nuclei isolation on other endpoints besides putting it into the single cell workflow, we could do a brief microscopy experiment. We could stain the nuclei and look at a 60x high mag objective microscope to check for blebbing. If we see blebbing, that indicates poor nuclei quality. If we are working with multiple samples, like we have a bunch of samples we need to do, it is best to prep the nuclei in small batches, perhaps 1 to 2 samples at a time, rather than prepping 8 samples of nuclei and putting all 8 through the single-cell workflow simultaneously. It may be best to do batches of 1 to 2 samples, get it to the first stopping step of the Illumina single-cell workflow, and then continue with more samples. And if we're doing that pilot study and we're looking under the microscope to do our nuclei optimization prior to putting them into Illumina single cell, we would hopefully see under the microscope smooth, uniform, high quality nuclei suspensions with these nice uniform round nuclei. But another method that you can take is doing total RNA isolation to do an RNA integrity number assay or a RIN assay with total RNA. If the RIN value or the RNA integrity number value of your nuclei isolation is below 6, it is likely to not be of sufficient quality to generate good single-cell data. So ideally, that RIN value should be greater than 6 if you wanted to directly test the quality of the RNA in your nuclei suspension. So now let's think about enrichment, and not just enrichment through different commercially available kits, but also through sorting, flow sorting of cells or nuclei. Many of those commercially available kits are using some sort of magnetic activated sorting, and all of these processes can be great for improving your viability, removing dead or damaged cells, or enriching for cell populations that you need. However, it can add a lot of time and stress to the cells and could potentially reduce the quality of the sample overall. So, doing pilot studies to optimize the speed, the pressure, and the runtime of your sort is imperative. And it might be necessary to include a live dead sort to improve or increase the viability of your cell suspension post-fax. So with those flow sorting recommendations, we want to make sure that we're using BSA instead of FBS. We know FBS is commonly used in cell culture, often used in flow sorting. We want to use BSA instead. FBS is a known inhibitory reagent in the Illumina single-cell workflow, so replace that FBS with BSA. We'll put up to 1% BSA in the tube, in the collection tube for blocking, and that should be more than enough. So for cell collection tubes, we would want to have 0.4 to 1 units per microliter of RNase inhibitor final concentration. And so an example of that would be we have a 40 microliter sort volume and we have a 40 microliter collection tube volume. That collection tube would start with 2 units per microliter RNase inhibitor so that when we finish our 40 microliter sort, we end up with 80 microliters at 1 unit per microliter RNase inhibitor. Nuclei collection tubes, same rule. We're just aiming for a higher concentration of RNase inhibitor and a max of 1% BSA final concentration. So that means if in this example you have a two-fold dilution value by your known sort volume, you might be able to start with 2% BSA in your collection tube. And the wash step. We're going to highlight the wash step here in a moment, but if you avoid all inhibitory reagents to Illumina single cell, then the wash step may be skipped at your own risk. The wash step is always recommended to do to wash out any potentially inhibitory reagents, but if you design your enrichment workflow to avoid all inhibitory reagents, it may be possible to directly go into the single cell workflow without doing this additional wash at your own risk. So what are some of these inhibitory reagents? Well, the one we've already mentioned was FBS or fetal bovine serum. And we've seen from internal tests that if even 0.1% FBS is in that final cell or nuclei suspension, or 0.0125% FBS in the PIP tube once you add your sample to the PIPs, it's going to cause low cDNA yield and low fragment sizes. It's going to mimic cDNA degradation. We don't think it's actually degrading the cDNA. There's some sort of like physical blocking that we think is the working mechanism here. But in either case, it's inhibiting the assay. It's reducing yield, it's reducing sizes, and so we want to avoid putting it into the assay. That's why that wash is so important. More than 1% BSA can interfere with breaking of the emulsions and emulsion forming and removing some of the waste steps. This can also result in in broken down mRNA that looks like degradation and low fragment sizes as well. High calcium ion concentrations, we just want to avoid those in the actual sample spike in because that could disrupt emulsion stability and the hybridization of mRNA. And organic solvents such as DMSO or methanol that are used in cryoprotective medias or fixation workflows, we want to make sure those are washed out sufficiently as well, since they can disrupt emulsion stability. And DNase-1 is frequently used in a number of tissue dissociations. So if you are using DNase-1 for your tissue dissociation, please make sure that you have gone through a sufficient number of washes of that cell suspension prior to putting it into the single cell workflow, because our barcodes and capture moiety is made of DNA oligos. So that DNase-1 will chew the PIP beads until they're shiny and bald. So we don't want blank beads. That's a bad time for everyone. And high molecular weight polymeric additives such as PBSA or DEPC. So these are frequently used to inactivate RNases, but that is fine to use for the downstream later molecular biology portions of the workflow where you need nuclease-free water and et cetera. But for the cell biology portion of the workflow where you're preparing your cells or your nuclei, we want to avoid using these particular molecules. Instead, just use RNase inhibitor instead of DEPC or PBSA. So what happens if you don't do that wash step or you do incorporate one of these inhibitory reagents? Well, here's an example tested by our development team where we used our Illumina single cell suspension buffer, and our cDNA trace comes out for a HEK293 cell line to look as is expected. When we use DMEM media only, we see a very similar phenotype for our cells. And then when we use DMEM media plus 10% FBS or PBS with 10% FBS, we see this smushed phenotype with reduced yield and a left-shifted average size mimicking cDNA degradation. So we want to make sure that we're avoiding this type of phenotype at the cDNA QC step with all of this left-shifted material. So Illumina single cell is incredibly flexible. It can capture any polyadenylated material, and a number of model sample tissue types have been reported to be successful from our customers. And so when we say demonstrated success, this means that one of our FASs or tech support staff has communicated with a customer that had intention to run these tissues and verbally communicated that they did it and they were happy with the results. That may or may not mean that they have published that study yet, and that may or may not mean that they shared their sample prep protocol with us at Illumina. So even if these samples have been demonstrated to be successful, that doesn't necessarily mean that us here at the support side have a step-by-step sample prep protocol for you for these sample types. That's always something that our customers are bringing to the table, and we are providing our recommendations to incorporate into those workflows. For non-model organisms, the same rules apply. Customers that have touched base with our FASs or our tech support staff that have been happy with the results from these species have also verbally reported success with all of these non-model organisms seen on the left here. And experiments that are ongoing, some of which may be successful by now, who knows, are on the right side of the slide here as well. So yeah, Illumina single cell is an incredibly versatile technology that we are excited to bring to all of you here today and talk to you about and answer your questions. And With Illumina single cell, one of the distinguishing factors with it is the ability to try generating your cDNA and library a second time from the same sample. So let's say something happened during library prep. I forgot to pipette mix a reagent and fragmentation wasn't efficient and we didn't get a good average fragment size or something like that. This would be for customers whose cDNA passes QC, but the final library for some reason doesn't. Accidents happen. So you could get the Illumina single cell supplemental enrichment and amplification kit, or the C kit, along with the Illumina single cell library prep module. This would give you enough reagents to regenerate your cDNA, that 15 soluble copy cDNA from your PIPS pellet that you store in -80, and try library prep a second time. And so with that in mind, that's something that is powerful for the Illumina single cell workflow. Is it's a powerful troubleshooting tool, and it is useful to know that there is a little bit of a safety net there, that if for some reason I spilled during the library prep, I can go back and generate cDNA. Now, this isn't a silver bullet for a sample that has insufficient sample quality, such as low viability or low RIN value going in. If you put garbage in, garbage is going to come out every time. So PIPseq replay, or this regeneration process, is not going to suddenly make degraded mRNA full length. So if the problem is further upstream, we have to troubleshoot and look further upstream at the sample prep, RNase inhibitor recommendations, and so on. And that is the end of